Hello, hello, ma. Checking. Bismillahir Rahmanir Rahim. I'm Dr. Muhammad Adil. Today, I will teach you recombinant DNA technology. Before talking about this technology, let me tell you about recombinant DNA, that what recombinant DNA actually is. It is actually, recombinant DNA is the name of a piece of DNA that has been engineered, combining at least two fragments from two different sources. This is actually an engineered piece of a DNA which has been obtained from two different sources. That piece of DNA is said to be recombinant DNA, and we prepared or engineered such a piece for a specific reason or a purpose, which is not available, but we want to prepare for some specific reasons, as we do for preparation of insulin. DNA molecules form by laboratory methods of genetic recombination, such as molecular cloning, that brings together genetic material from multiple sources, creating sequence that would not otherwise be found in the genome. So creating or engineering a sequence which is not already available is said to be the recombinant component or complement. Second, recombinant DNA molecules are sometimes called chimeric DNA. Because why are they called chimeric? Because sources of such DNA, these are different. The reason it is called chimeric DNA. The DNA sequence used in the construction of recombinant DNA molecules can originate from any species. This is very much interesting thing, that we can obtain DNA from any source. It can be from plant, it can be from bacteria, from human DNA, and we can join it with any of the DNA. So this is quite astounding, and actually the advent. In addition, DNA sequence that do not occur anywhere in nature may be created by the chemical synthesis of DNA and incorporated into recombinant molecule. Now let's talk about technology. If any of you would have been there, I would have surely asked you about the term technology, that what actually technology is. Technology is any prescribed procedure or designed way where we engineer things of our own wish and will, where things are prepared according to our own desire, where required things, where required materials of any nature, they are being prepared. The type of procedure is said to be technology. Here we are talking about recombinant DNA technology. So this is a kind of technology in which we prepare DNA of our own will and desire, where we engineered DNA segments which are required to us. Using recombinant DNA technology and synthesis DNA, literally any DNA sequence may be created and introduced in any of a wide range of living organisms. And this is, I think, the quite astounding achievement of this era. And this is a very huge advancement in the field of genetics. Recombinant DNA differs from genetic recombinations, and the former result from the artificial method and the test tube, while the latter is a normal biological process that result in the remixing of existing DNA sequence and essentially all organism. Now, what we do, we actually prepared a segment which is required to us, So this is a kind of artificial things which we do outside the living body. And a test tube, we engineered a segment. I'll thoroughly discuss all those procedures and steps in detail, that how recombinant DNA is formed. What are the factors responsible for preparations and management of this recombinant DNA? We will talk about all those factors and components and details. But let me tell you, this is actually the segment of DNA which we engineered. And then so there are two way of getting recombinant DNA, and then we utilize it for our desired purpose. The first thing is that we prepare it artificially outside a living body. We provide a culture for preparation of this segment which is desired or which is required. And then what we do, then we transfer it to a living body, wherein this segment simultaneously it redivide, and we got a number of such segments which we then use for specific purposes, for the purposes which we have prepared or engineered that segment. So there are two, we can say, areas where it works, at preparation works. The first one is artificial, and the second one is the body of the organisms. Okay, recombinant DNA is a form of DNA constructed in the laboratory. This is what we prepare in laboratory. It is generated by transforming selected species of DNA from one organisms. Here, two organisms are involved. You can see in the picture, there is a vial, and there, if you focus it, insulin is written on it. So from two different sources, we select DNA. We prepared DNA or segment of our own will, which encompasses different kinds of codes which we needs, which we requires, which enable us to get the very specific thing which we required. And what happened then? So there is this is actually a very challenging thing. Its example is insulin. Insulin is a life-saving thing, I would say. And this is due to the technology, which has quite challenging factors. Insulin can linger on survival, though from religious point of view, it has been already fixed that who is going to die when. But scientifically talking, insulin is playing a vital role in saving lives. So these are all actually the fruits of this technology. It is now, you can see on the screen, the vial shown in the photograph contain human insulin, one of the first therapeutic protein that was genetically cloned. The drug is used to treat diabetes. Now, let's talk about the restriction modification system. What is this system? What are the significance of these systems? What is the utility of this system? And what role it actually plays in formation of that segment which is required? And what is the basic role of restriction modification system in that very specific technology, which is called recombinant DNA technology? The restriction modification system is found in bacteria and other prokaryotic organisms and provide a defense against foreign DNA. What do it do? It is found in bacteria and also other prokaryotic organisms. Bacteria have restriction enzymes, and we will talk about all the enzymes which are being used in this technology and which play a pivotal role in this technology and the development of this technology. And actually every step of this technology basically based on these enzymes. So the very first and the most important enzymes, which is known as RE. RE mean restriction in endonuclease. It is enzymes which cause cleavage in double-strand DNA. It's different specific points. This prevent infection by effectively destroying the foreign DNA introducing by infectious agents. So endonuclease actually it works as cleavage. The segment which we required and the sequence of the DNA segment where there is thymine, adenine, guanine, cytosine, and then sequence of all these bases. We cleave DNA at certain positions. And what do it do? It actually, this is endonuclease enzyme which cut the DNA at that specific points. Now let's talk about the types or classification system of restriction modification system. The type 1 system are the most complex, consisting of 3 polypeptides: R restriction, M modification, and S specificity. The resulting complex can both cleave and methylate DNA. Both reaction requires ATP, adenosine triphosphates, and cleave often occur a considerable distance from the recognition sites. The S subunit determines a specificity of both restriction and methylation. Cleaves occur at variable distance from the recognition sequence. So discrete bands are not easily visualized by gel electrophoresis, which is a technique wherein we can observe. This is another system and the most commonly used system in this technology because it is very much accessible. This is simple and most prevalent. Instead of working as a complex, methyl transferase and endonuclease are encoded as 2 separate proteins and act independently. There is no specific protein. Both protein recognizes the same recognition site and therefore compete for activity. The methyl transferase act as a monomer, methylating a duplex unstained at a time. The endonuclease act as a homodimer, which facilitate the cleave of both sides. Cleavage occurs at a definite positions close to or within the recognition sequence, thus producing discrete fragments during gel electrophoresis. For this reason, type 2 systems are used in labs for DNA analysis and gene cloning. This is the type 2 which is used for DNA cloning worldwide, and this is the most accessible one. Type 3 system have R and M, restriction and modification proteins that form a complex of modification and cleavage. Here, you should not be confused with this word modification because the already available DNA, we actually modified that DNA. We actually, what we do, we modify the sequence of that DNA, and every sequence of DNA is responsible for the production of a specific type of proteins, which in turn do its job, which has been assigned to that very particular segments. So if preparing a sequence of segment would mean that there are present some codes which will enable that sequence to form proteins, and in terms what will it do? It will do its specific work for which the segment has been engineered. The M protein, modification protein, however, can methylate on its own. Methylation also only occurs on one strands of the DNA, unlike most other known mechanisms. The heterodimer formed by the restriction and modification proteins competes with itself by modifying and restricting the same reaction. This results in incomplete digestion. Now, let's talk about the type 4 system, they are not true restriction modification system because they only contain a restriction enzyme and not methylase. Unlike the other types, type 4 restriction enzyme recognize and cut only modified DNA. So here we can work on the already modified DNA and we can cut pieces according to our own will. Enzymes. Now let's talk about the enzymes which are present and which play roles in this technology. The first and the most important one is endonuclease, which cleave the terminal phosphodiester bonds. They do so only often recognizing specific sequence in DNA, which are usually 4 base pairs long and often palindromic. Now, one thing which you must focus that the sequence of the DNA must be palindromic. And before going towards palindromic, you must know that what actually palindromic is, which can be read from the same from either side. If we want to read it from left to right or right to left, it may looks like same. For example, madam, M-A-D-A-M. If we want to read madam from either side, it will, the spelling of both will be same. Like write on a page this word madam and read it from either side, it will be pronounced the same, madam. Palindromic figures are there in order to make it more simple so that you will understand that what palindromic actually is. Write 11. So from either side, it will be 1 or 22 or 33 or 66 or let's suppose 6666. So if you are 8888, 9999, from either side, if you want to read it, it will be same. So this mean palindromic. So like that, the sequence of DNA which will be cleaved on either side, it must be palindromic. The first condition. Okay, restriction enzymes, also called restriction endonuclease, a protein produced by bacteria that cleaves DNA at specific sites along the molecule. This is restriction enzymes, and it is also known as restriction endonuclease because it cleaves the DNA on either sides. In the bacterial cell, restriction enzymes cleave foreign DNA, thus eliminating infecting organisms. What it do? It actually cleave the foreign DNA. It cleave at specific points, and through these enzymes we can get a very specific segment which is required. Restriction enzymes can be isolated from bacterial cell and used in the laboratory to manipulate fragments of DNA, such as those that contains genes. For this reason, they are indispensable tools of recombinant DNA technology. So this is actually the very first component of this technology. Through this restriction enzyme, we can commence this technology and engineering of desired DNA. This is the diagram. You can see it, though it's in a landscape way. You can read it. Second, DNA ligase. Here is written 1, but it is actually the second enzyme. DNA ligase, it is, we can say, antonym of the first one. The first one work as a cleavage. The first one, what the first one do, it cleaves the DNA at specific points. And what DNA ligase do, it actually ligate both of them, the cleaved parts. It actually ligate the cleaved parts. So DNA ligase is isolated from E. coli and bacteriophages commercially and is used in recombinant DNA technology. These are the sources which you came to know about from Escherichia coli and bacteriophage. We can obtain these enzymes. And DNA ligase, actually what it do, it joins the DNA fragments which were once cleaved by the restriction endonuclease enzymes. And what it do, it joins the DNA fragments with cloning vector. Vector, it is actually the carrier of the DNA. One is foreign one and the other one is vector. We will talk about this vector in detail. Reverse transcriptase, third one. Reverse transcriptase is used to synthesize complementary strains from messenger RNA template. Used what it do, it makes it prepared complementary strains from messenger RNA. It is also known as RNA dependent DNA polymerase because this has been formed using messenger RNA. It is isolated from retrovirus. Restriction endonuclease, again, we have talked about it in a detail, but here we will discuss three types of endonuclease enzymes which cause cleavage and through which we can get the required template, the required segment of DNA, which in turn produce the required segment and proteins which functions what we need. Okay, the first type is restriction, type 1 restriction endonuclease. It has both methylation and endonuclease activity, as we have told before. It causes modification. There are two. This is we would say biodisciplinary. It play a biodisciplinary role. Its function is biodisciplinary. The first one is methylation and then endonuclease activity. It requires ATP to cut the DNA. This energy is required in cutting the DNA segment. It cuts DNA about 1,000 base pairs away from its restriction site. So this is one of the distinction, that it cuts DNA at 1,000 base pairs away from its restriction site. Type 2 restriction endonuclease, it does not require ATP to cut DNA. Now this is the variability which we can see. It cuts DNA at restriction site itself. What it do? It at restriction site it cut the DNA. Type 3 restriction endonuclease, it also requires ATP to cut DNA, and it cut DNA at about 25 base pairs away from restriction. What is the difference between the first and the third type of restriction endonuclease? You must remember, and this is we can say it's a very important from MCQ's point of view. You may have it in exam. So type 1 is 1,000 base pair away, while the type 3 is 25 base pair away from restriction site, and type 2 is it cuts DNA at exactly the restriction site itself. The fourth one is terminal transferase. It is the enzyme that converts blunt end of DNA fragments into sticky end. Why our end should be sticky? Because if it is sticky, it will be able easily ligate it. And if it is not sticky, the ligation will be difficult. If the restriction enzyme cuts DNA forming blunt ends, then efficiency of ligation is very low. So the enzyme terminal transferase converts blunt ends into sticky ends. This is a very important role, and this is supposed to be very considered very important, because there are chances of making blunt ends and they're actually generated DNAs at having blunt ends. But if we do have terminal transferase, so what it do, it make it feasible to be ligated. So because what it do, it converts the blunt end into sticky ends, which become easy for ligation. Terminal transferase enzyme, what it do, the second function, it synthesizes short sequence of complementary nucleotides at free ends of DNA so that blunt ends is converted into sticky. This is actually the physiology, or we can say a bit short mechanism how it makes that end sticky. Here is written fifth, but it is actually the sixth one, nuclease. The enzyme nuclease hydrolyzes the phosphodiester bond on DNA strands, creating 3 hydroxyl groups and 5 phosphate groups. It usually cuts DNA on either side of distortion caused by thymine dimers or intercalating agents. The gap is filled by DNA polymerase and strand is joined by DNA ligase. Nuclease are of two types, endonuclease and exonuclease. DNA polymerase. DNA polymerase is a complex enzymes which synthesize nucleotides complementary to template strands. It actually adds nucleotides to free hydroxyl ends and help in elongation of strands. It helps in elongation of strands by adding hydroxyl group. And one thing more you must know, phosphodiester bond, it actually play a vital role in this technology. It adds nucleotides to free 3 hydroxyl ends. Okay, it also helps to fill gap in double-stranded DNA. DNA polymerase 1 isolated from Escherichia coli is commonly used in genes cloning. Okay, now let's talk about ribonuclease. Ribonuclease removes messenger RNA from DNA. RNA heteroduplex, and that messenger RNA is used to synthesize complementary DNA. It is isolated from retrovirus. Alkaline phosphate, the enzyme phosphatase, with every enzyme is, if it should be at the end because I pronounce it phosphate. So if it is phosphate, then we cannot call it enzymes. For an enzyme, it is the identity that it should be written as at the end. Alkaline phosphatase. The enzyme alkaline phosphate helps in removal of terminal phosphate group from 5' end. It prevents self-annealing of vector DNA soon after cut open by restriction endonuclease. Polynucleotide kinase. It adds phosphate groups from ATP molecule to terminal 5' end after phosphorylation by alkaline phosphate. Now, I would like to discuss with you all these outlines, which we will talk about in our course. Though this course is confined and it's not that lengthy as systematics. This is, I think, second semester. So you are second semester students, so systematic is not your subject. But in systematic, there is a longer and quite lengthy course, and this is a bit confined. We will read things to the point, and we have covered almost 3 topics of our contents. And let me conclude it a bit. I'm going to start it from the first so that you may know. You can say it's a kind of summary. So if you heard it, if you will listen it, you will understand the whole story, which we talked about. We actually talked about the complementary recombinant DNA, and then we talked about technology, and then classification and all the types of that restriction modification system. Then we talked about the different kinds of enzymes, and these were actually the enzymes which are responsible for the accomplishment and amplification of recombinant DNA. Now, Sorry, 1 minute. Now let me tell you what we are going to read, and what are the course outlines. We talked about the destruction and modification system, types, enzymes, genetic and application, nomenclature, cloning vectors, plasmids, viruses, phages, cosmids, and phosmids, cutting and joining of DNA molecules, which we already discussed, isolation and purification of DNA, cuttings of DNA molecules, ligation of DNA molecules, blunt ends and cohesive termini, homopolymers, tailing, cloning strategies, selection and characterization of recombinant molecules, verification and amplification of desired genes, gene bank, PCR, RFLP, DNA sequencing techniques developed for cloning eukaryotic genes and prokaryotes, host systems available like bacteria, yeast, plant cell, animal cells, applications of genetic engineering in medicine, agriculture, environment, and molecular biology. These would be the application which we have talked at the end. As I said, genetic engineering and medicine, agriculture, environment, and molecular biology, these are actually the utility of these techniques. So we will talk about that in the last. Now let me tell you what we said. We said recombinant DNA refers to the creation of new combination of DNA segments that are not found together in nature. So what we did, we isolate and manipulate genes, allow for more precise genetic analysis, as well as practical applications in medicine, agriculture, and industry. Now then, how to make recombinant DNA? First, what we did, we have to isolate DNA, then cut with restriction enzyme, then ligate into cloning vector. And after that, transform recombinant DNA molecule into host cell. What will happen after that? Each transformed cell will divide many times to form a colony of millions of cells, each of which carries the recombinant DNA molecule, and that what was required. Now, this was actually the whole story, which we will discuss later in detail, each and every steps. Thank you. Now this is the end of the lecture. The later or rest of the course outlines we will talk about in next lecture, inshallah.